Hello students, welcome back to my channel Learn Biology by Pravina. In my previous video, I asked question and I told that whoever is going to give the answer in the comment section first, that person name I'm going to take. So the name of the person here is a Legend Hodi Baba. So very good. So you are the one who gave the answer in my comment section first. So question for today's lecture. Why DNA is called polynucleotide molecule? So student, those who know the answer, please give your answer in the comment section. So in this today's video, we are going to see the topic that is packaging of DNA helix. So in the previous lecture, I told that why the DNA need to be get packaged, because the length of the DNA is much higher because it has to get entered into the nucleus, so the DNA has to get packed. So now let us see how in case of prokaryotes the DNA is being packed. So in case of prokaryotes, so like if you take an example of E. coli, so we all know that DNA is negatively charged. So now this negatively charged DNA held with some positively charged protein to form nucleoid. So you can see here in the diagram. So this is the nucleoid. So now this DNA is being held together by what? Positively charged protein and results in the formation of nucleoid. So this is how the DNA is being packed in case of prokaryote. So now let us see how the DNA is packed in case of eukaryotes. So the packaging of DNA in case of eukaryote, so we all know that eukaryote, the organism which has well-defined nucleus. So how this DNA is being packed in case of eukaryote? So in case of eukaryote, there is a set of positively charged proteins that we call it as histone. Now what are these histones? Now these histones are rich in positively charged basis amino acid reduce, that is lysine and arginine. Okay, so now this there are 8 histone that forms a histone octamer. Okay, so 8 histone that are held together and forms a histone octamer, and this negatively charged DNA wrap around this histone octamer to give a nucleosome. So you can see here, so this is the actually the structure, this is the histone protein actually, and this is the DNA that has been wrapped around the histone protein. Okay, and results in the formation of structure that we call it as nucleosome. So here in the diagram we can see here, so this is the structure of the nucleosome that has been formed with the what? 8 octamer. Okay, so you can see here these are the histone protein, that is how many protein you can see, H4 here, H3, 2H4 and 2H3 and H2B and H2A. So these are the 8 protein that are held together and results in the formation of histone octamer. And when the DNA is wrapped around this histone protein, then with this structure we call it as nucleosome. So here in case of eukaryote, a typical nucleosome contain around 200 base pairs. So what does it mean? Suppose this is the nucleosome, and in this nucleosome a DNA is being wrapped. Now this DNA, how many base pair it constitute? Around 200 base pairs. So this DNA or nucleosome constitute of 200 base pairs. So similarly we need to find out the total number of nucleosome in case of human beings. So how can we find out this one? That is, we all know that is normally we have how many base pair? That is 6.6 into 10 to the power 9 base pair. So that has been divided by 200. Then you will get the answer as 3.3 into 10 to the power 7 nucleosome. So in case of eukaryotes, so how many nucleosome we possess? That is 3.3 into 10 to the power 7 nucleosome that is present in case of the human being. Now this nucleosome constitutes a repeating unit to form chromatin, and this chromatin is the thread-like stained body, and the nucleosome in chromosome is actually look like beads on this string. So you can see in the diagram, actually this is the DNA, okay, double helix model of the DNA. So this is the DNA, and this DNA is being wrapped around histone protein and results in the formation of nucleosome. Now this nucleosome is at this stage, it looks like a beads on this string. Now it's again keep on coiling, so that it results in the formation of chromatin fiber. Chromatin fiber further coiled and results in the formation of chromosomes. So at the end, the structure that we see in the microscope is a chromosome. So basically in the nucleus you might have seen thread like structure like chromatin fiber and chromosome. So they are nothing but these are the double helix DNA that has been coiled around the around the nucleosomes. So now we are going to see the packaging of DNA in case of eukaryote. So chromatin is packaged, results in the formation of chromatin fiber. This chromatin fiber coiled and condensed at the metaphase stage and results in the formation of chromosome. So you can see in the picture here. So this is actually the double helix model of DNA, right? So you can see in the diagram here, this is the double helix model of DNA, and in the next stage it's look like beads on this string, and this is the chromatin fibers. This chromatin fibers is further extended and they keep on condensed, and at last it looks like a chromosome. Got it, students? So this is how, that is why whenever we see the nucleus in the microscope, the DNA basically looks like a chromosome. So this chromatin has two forms. So they are euchromatin and heterochromatin. Now what are euchromatin? Euchromatins are nothing but these are loosely packed transcriptionally active region of chromatin. So it is stains lightly. And what are heterochromatin? These are densely packed inactive region of chromatin. It's stained dark. So when you see the diagram, so here you can see these are the loosely packed, that is euchromatin, and it is what stained lightly, and these are tightly packed heterochromatin and it stains darkly. Okay, students, so hope you all understood how the DNA molecules are being packed in case of prokaryote as well as in eukaryote. So thank you for watching this video. Don't forget to subscribe this channel, then only you will get my video as soon as I'm posting the videos. If you have any doubts, please ask your doubts in the comment section. So thank you for watching this video. Thank you, students.